Okay, students, so very good afternoon to all of you. So let's continue with my section of last discussion of composting. So as you all know, today we were in a position to describe the substrates for composting. Students, we all are now very much aware of the fact that how beneficial composting is. That is, they are able to provide the nutrients and they have a much more effective phenomena as compared to the chemical fertilizers. So this diagram, this picture will be able to assist you that how important the role of microorganism is in carrying out the degradation process of all those raw materials that is actually acting as a substrates for composting. So dear students, you should know that these substrates are belonging to different chemical origin, like carbohydrates, like certain other components of plants such as lignin, cellulose, hemicellulose, pectin that you have already studied in your BTY 153. So let's talk the detail part of that. So there are certain number of substrates which are being decomposed by those microorganisms in different phases, like fungi, like bacteria, like actinomycetes, etc. So if I talk about lignin, so you all know that it is a structural component of plant and has the ability to be degraded very slowly. But its degradation, who is causing its degradation? Which are microorganisms? It is fungi. Such fungi are also called as white rot fungi. You should remember the names like Tramites or Sterium hirsutum. So don't forget the name of these fungi. So I am again repeating that lignin decomposition is carried out by fungi. And then cellulose, you also know it is a most abundant part of the cell wall of the plants. So cellulose is actually built up of repeating units of glucose. So when cellulose is being degraded, it has to be degraded into three different steps. First step is to cleave that beta 1,4 bonds which are available in cellulose within the molecule. So it is being degraded by endo beta 1,4 gluconases. I mean, it's very easy. If you remember glucose, you can remember the enzyme, right? Gluconases. Then next is exo beta 1,4 gluconases, which separates those cellobios. Cellobios is formed after the first part of degradation, that is from the endogluconases. Now, who is going to degrade cellobios? It is beta glucosidases. It hydrolyzes the cellobios and convert it into glucose, which is taken up by the microorganism. So see, first cellulose was very complex, so it is being degraded with the help of these three different enzymes. Now after the at the end, when you got the glucose, it is basically being utilized by the microorganisms. And now let's see microbes which are involved in cellulose degradation. I am repeating, in lignin it was fungi, but here in this case it is fungi and bacteria both. Which fungi? It is, if you remember easily, AF, Aspergillus and Fusarium. Bacteria, Pseudomonas, you always remember. Cytophaga is another one. Two is okay, two examples is fine for both categories. So do remember students that we do have the process for aerobic and anaerobic condition. For aerobic, it is the bacteria and fungi which degrades mainly, and for anaerobic it is Clostridium. Okay. Next is hemicellulose. So hemicellulose is have different components. Xylem is one such component of hemicellulose, and it is found in straw, bagasse, etc. And what is the enzyme which are involved primarily over here is xylenases, right? So from the word xylen, enzyme degrading, it is xylenases, which is produced by bacteria and fungi, right? Then the next category is pectin. Fine. So it is degraded by pectinase, which is quite commonly found in fungi and bacteria. And then starch. Okay. So all these you must have studied in carbohydrate section, in polysaccharide section in your BTY 153. Even then, if you have any confusion, any doubts, I will be available on Zoom session from your 2 to 3 p.m. today's class. So starch is degraded by enzyme named as phosphorylase and amylase, right? So let's talk about the see, these were the substrates and these were the microorganisms. Now the phenomena, right? So you should see that the microorganisms are converting it at a very good rate, at a very high density, right. So metabolic activity of microorganism is quite good in this phenomena. So there will be a constant change in certain conditions like temperature, pH, moisture. So you know, from the raw material till the compost formation, there are regular changes in these components. And if I talk about the number of phases, composting is basically divided into 4 different phases. Phase 1, which is Mesophilic phase, I have described this in the last slides. The temperature range of this phase is from 25 to 40 degrees centigrade, and this is actually the starting phase and very rich, energy rich stage, right. Here you will see that the compounds are easily degradable into means like easily degradable like sugars, like proteins, and they are easily being degraded by fungi, archaebacteria, actinobacteria and bacteria, right. So these are the primary decomposers. Now compost worms, mites, millipedes, etc., they are also developed at this stage during this primary decomposition stage. So slowly and slowly metabolic activity of these components raises the temperature. See how easy it is to understand. First, the temperature range which I have mentioned over here is 25 to 40 degrees centigrade, but since the metabolic activity of microorganisms start, it causes the rise in temperature, and this rise in temperature is making it to reach to the next phase, that is thermophilic phase, right. So now the organisms has to understand, they have to adapt at this temperature so that they could get an advantage, and they should replace the mesophilic microbes, right. So the decomposition here continues to be fast, and it is reaching you know, it is accelerated at a very high rate. So obviously if it happens, the temperature will rise, and something about 62 degrees centigrade reaches. So you should understand that in this phase the microorganism, which is a thermophilic fungi, has to show the growth, maximum growth, right, in this range. So but more than this temperature, the fungal growth will be inhibited. Okay. So at this stage, at this temperature, most of the thermophilic bacteria and actinobacteria remain active, right. So even then at that temperature range, destruction of certain microorganisms occurs. Temperature even keeps on rising because of metabolic activities, and it may exceed 80 degrees centigrade, and because of this exothermic reactions, it reaches to that temperature. But you know, because of that, you know, regular turning up of the pile is required so that temperature should be uniform over there. So this phase must be clearly understood. Thermophilic phase is very important for pasteurization, also to kill pathogens, weeds and insect larvae. Okay. So third phase is cooling phase, because now the temperature has been so high. So here the activity of all those thermophilic organisms is stopped, and exhaustion also takes place, that is all the substrates, temperature, you know, all the substrates gets exhausted. जब आप constantly किसी चीज़ पर act करते हो तो substrate is going to be exhausted, right. So and now temperature starts to decrease. Fine. So mesophilic organisms now come back again. They are trying to recolonize the substrate. अब ये कहाँ से आ रहे हैं? Maybe they were having certain spores where they survived themselves, and from external inoculation also, right. So in this second phase, the mainly the degraders are starch or cellulose degraders. Now the last phase is maturation phase. What happened is that here the quality of substrate declines actually, and composition is entirely altered, right. So fungi increases over here, while the bacterial number gets declined. So compounds which are not further degradable, such as lignin humus complex, are formed and becomes predominant. So as I talked in the last slides, also in the last presentation of last lecture, I told you that humus is being mainly found, which is actually acting as a soil conditioner. So this complex is further non-degradable. So this lignin humus complex is being formed that provides the conditioning to the soil. So microorganisms categorizations are being described in the coming slides. You have to remember the name of microorganisms such as Bacillus, Pseudomonas, etc. Fine. So actinobacteria, several actinobacteria which are thermotolerant, which can understand the temperature range. Then thermos group, you have to remember certain names. Then archaea is also helpful. They are thermophilic, right? So why they are low in numbers is mainly because they are oligotrophic and their generation times are much higher than those of bacteria, right? So that is why they are not suited for changing condition. Composting is a regular changing condition. Okay, you also got fungi. So fungi and bacteria actually competes with each other. So bacterial growth rate is higher than fungi. You can understand that, and that is why fungi are very easily being out competed, while good supply of oxygen is also more important for fungi than for bacteria. So due to these reasons, lower thermotolerance, fungi plays a negligible role during the thermophilic phase. Okay, so you can understand the difference between the bacteria and fungi. So dear students, apart from this, I'll be also discussing some parts of biofertilizers in the coming lecture. So I'll be available on Zoom from 2 to 3 p.m. I have already shared the meeting ID and password for that. I hope most of you students needs to be available for that session. I'll be interacting live with you to discuss all these things once again. Thank you.